Welcome to the Deep Dive, where we take the essential information from the most complex scientific topics and distill it into immediately useful knowledge for you. We're going microscopic today, focusing on one of the most critical, yet often unappreciated battles happening inside every single cell in your body, the constant struggle for protein quality control.
Yeah, a really fundamental process.
Our subject is the endoplasmic reticulum stress, stress, and the brilliant, sometimes brutal, mechanism cells use to cope with it. That's the unfolded protein response, or UPR.
That's right.
If you've ever encountered research about chronic illness, think Alzheimer's, cystic fibrosis, type 2 diabetes, and heard the term protein misfolding, well, you're about to get the full story on the sophisticated cellular system behind it.
It really is at the heart of so many conditions. Okay, let's unpack this. We often use this analogy. The cell is a high-tech factory, a good one.
If that's true, then the endoplasmic reticulum, the ER, it's this enormous networked assembly line, packaging plant, and quality control center. It handles pretty much every protein destined for secretion or for placement on a membrane.
Thousands of polypeptide chains synthesized there every second. It's incredibly busy.
And they absolutely must be perfectly folded into their specific three-dimensional shapes to function correctly.
Yeah, form dictates function, always in biology.
So what happens when the demand just overwhelms the capacity, or the products start coming out faulty, and the whole system, locks up? That catastrophic breakdown, that's ER stress.
Exactly. And if we connect this to the bigger picture, understanding the UPR is absolutely crucial because, well, it really dictates cellular survival across every tissue type.
It's that fundamental. It is. The ER's core function is non-negotiable. Folding these newly synthesized polypeptides, often it gets help from molecular assistants we call chaperones, right? The helpers.
But misfolding, it happens. It's a natural occurrence, especially with really complex proteins. The problem arises when those misfolded proteins persist, when they accumulate. That's the definition of ER stress.
Accumulation is the key.
Yes. And the UPR, the unfolded protein response, that's the cell's universal synchronized attempt to restore balance, or what we call protein homeostasis.
So it's an adaptive mechanism first, trying to fix the problem.
Primarily, yes, it's designed to triage the crisis. But, and this is critical, if that triage fails, the UPR doesn't just, give up. It shifts strategy entirely. It actually becomes the agent that initiates programmed cell death, apoptosis.
Wow. So it tries to save the cell, but if it can't, it triggers the self-destruct sequence.
Precisely, a controlled demolition to protect the organism.
Okay. So our mission today is to explore the specific triggers that push the cell into this panic mode. We need to detail the three distinct molecular pathways the UPR uses to fight back.
The IRE1, PERK, and ATF6 pathways.
Right, get into the mechanics there. And finally, connect this microscopic quality control failure to the massive clinical relevance it holds across chronic disease, including, some of the newest therapeutic strategies being tested.
Sounds like a plan. It's a fascinating system.
Let's kick off at the source of the problem then. What actually initiates this quality control crisis? You said accumulation, but what causes that?
Well, the core principle is simple, really. The folding demand has exceeded the cell's folding capacity.
The assembly line is jammed.
Pretty much. We can group the causes into, say, 4 major categories. First, and maybe the most fundamental, is the issue of inherently flawed raw materials. Let's call these genetic factors.
Yeah.
If you have a deleterious mutation, a harmful change in the gene sequence encoding a protein, the resulting polypeptide chain might be physically incapable of folding correctly. Or maybe it folds, but just incredibly slowly.
So the problem is baked into the design from the start.
Exactly. No matter how many chaperones you throw at it, the basic structure is compromised. We see this starkly in diseases like cystic fibrosis. Yeah, the CFTR protein, due to common mutations, has a design flaw. It's inherently prone to misfolding. This isn't really a problem of the ER's capacity, it's a problem of protein design. The cell has to deal with products that are genetically defective right off the assembly line, placing this unremitting burden on the ER.
Okay, so that's one cause. What else can jam the system?
Well, then you have the sheer volume problem. We can call it capacity overload or high demand.
Right, like the factory is fine, but it's asked to do way too much work.
Perfect analogy. This is where the cell itself is intrinsically healthy, but its normal job suddenly requires a massive, sustained output of proteins.
Can you give an example?
Here, think about specialized cells that are essentially protein secretion machines, like plasma cells making antibodies to fight infection.
Okay.
Or very relevant metabolically, pancreatic beta cells churning out insulin. In insulin-resistant states, like early type 2 diabetes, the body demands far, far more insulin than usual.
Ah, because the tissues aren't responding well to it.
Exactly. So that chronic, heightened demand forces the beta cell to synthesize vast quantities of insulin. The sheer volume of new polypeptides entering the ER just overwhelms the folding machinery, even if that machinery is working perfectly fine.
It's like demanding a 5-fold increase in output from your factory overnight.
Yeah, the quality control staff, the chaperones, they simply can't keep up with the influx. You get an immediate backlog of misfolded proteins.
Makes sense. High demand. What else?
A similar kind of overload, perhaps even more acute, happens during infection overload, especially viral infections.
Oh, so.
Well, when a host cell is infected by a virus, the virus hijacks the cell's ribosomes, its protein-making machinery, to synthesize viral proteins, structural proteins, replication proteins.
Ah, it forces the cell to make enemy components.
Right, and this sudden, massive, often uncontrolled flood of new, often non-native polypeptides just swamps the host cell's ER. The cell now has to try and manage its normal folding load alongside this huge influx of viral stuff. That triggers ER stress, partly as a defense mechanism against the invader, too.
It sounds like a truly stressful environment inside that cell. But you mentioned external conditions, too.
Absolutely. The environment inside the cell can definitely impact the quality of the fold. This covers what we can call environmental and metabolic insults.
Okay, like what?
Well, protein folding is highly dependent on specific chemical conditions. A major factor is the cell's redox state, the balance between oxidizing and reducing conditions.
It's redox state.
Yeah. The ER is actually engineered to be a highly oxidative environment compared to the cytoplasm. There's a reason for that.
Arch is.
It's because forming crucial disulfide bonds, those sulfur bridges that really lock a protein's tertiary structure in place, requires an oxidizing environment.
Ah, yeah.
So if the intracellular redox state shifts, maybe due to metabolic stress or some toxin exposure, the enzymes responsible for creating and isomerizing these disulfide bonds, like protein disulfide isomerase, PDI, get compromised.
It can't work properly.
Right. And if you can't form stable disulfide bonds efficiently, folding efficiency just plummets. Misfolded proteins pile up.
So the structure literally can't set properly if the internal chemistry is off.
Precisely. And the final critical stressor, maybe the most common in acute situations, is energy deprivation.
Lack of energy, like ATP.
Exactly. Protein folding isn't passive, it's actually quite energy intensive. Many of the key players, the foldases and chaperones like BiP, which is a major ER chaperone, also called GRP78, they need ATP.
Why do they need ATP?
They use its energy to bind to and release polypeptides repeatedly. It's like they're using energy to nudge and guide the protein through the complex folding process.
Got it.
So if the cell suffers from hypoxia or ischemia, lack of oxygen or blood supply, like in a heart attack or stroke, ATP production just collapses.
Right. No oxygen, no efficient energy production.
And when ATP levels drop precipitously, the power supply for the chaperones is effectively cut. The folding machinery grinds to a halt. This dramatically increases the burden of misfolded, often sticky proteins that can then clump together or aggregate. This is probably the most ubiquitous and immediate trigger for acute ER stress.
Okay, so we have genetic flaws, overload from demand or infection, bad environmental chemistry like redox shifts, and lack of energy. All roads lead to a jammed ER.
Essentially, yes. Too many unfolded or misfolded proteins accumulate. So the ER is jammed, the alarm bells are ringing. Now we get to the core of it. The unfolded protein response. You said it's not one action, but like a coordinated operation.
That's right. It's a highly coordinated response utilizing 3 distinct yet interconnected signaling arms or pathways.
And these are controlled by sensors.
Yes.
Yes.
The UPR is managed by 3 key transmembrane sensor proteins. They sit embedded in the ER membrane. In a healthy, unstressed cell, these 3 sensors, their names are PERK, IRE1, and ATF6, are kept quiet, kept inactive.
Oh.
They're physically bound to that master chaperone we mentioned, BAP, binding immunoglobulin protein. PP essentially keeps them leashed.
Okay.
But when misfolded proteins start to accumulate in the ER, they need chaperoning. So they start binding to BiP, effectively pulling BP away from the sensors. So the presence of the misfolded proteins frees up the sensors. It's like they act like a wrench that pulls the safety pin off the grenade.
Huh. That's a great way to put it. The binding of BP is the safety switch. When BP gets preoccupied tending to the misfolded protein emergency, the three sensors are released. They then typically pair up dimerize, and this activates their enzymatic functions on the other side of the membrane in the cytoplasm.
Activating all three response pathways more or less simultaneously. Generally, yes, though the kinetics and downstream effects differ. Let's start with the pathway that initiates perhaps the most critical immediate defensive action. The PERK pathway, PERK.
Okay, PERK stands for PKR, like ER kinase. When it's activated, when BP lets go, PERK dimerizes in its kinase domain, immediately phosphorylates a specific substrate called EIF2A.
EIF2, what does that normally do?
It's a crucial component of the machinery that initiates translation. That's the process of synthesizing proteins from mRNA blueprints.
So PK phosphorylates it, which means
By phosphorylating EIF2, PRK effectively puts the brakes on global protein synthesis. Most translation stops. That's action 3. Slow protein translation. Exactly. It's the cell hitting the immediate emergency start button. Slow down the influx of new protein products into the already jammed assembly line. It gives the cleanup crew, the chaperones, and degradation systems a chance to catch up.
that makes perfect sense as an immediate triage step. Cut the supply line. But stopping all protein production, that can't be good long-term, can it? You're right, it's definitely a double-edged sword. It's a temporary measure. But here's the subtlety. While global translation is inhibited, the phosphorylation of EI2 actually selectively enhances the translation of certain specific mRNAs. Which ones?
Most notably, a transcription factor called ATF4. The cell manages to specifically translate the ATF4 message, even when most others are shut down.
And why is ATF4 important?
ATF4 is critical because it travels to the nucleus and drives the expression of genes involved in amino acid metabolism, antioxidant responses, and importantly, it helps ramp up the production of components needed for the other UPR branches and for eventual cell death if things don't improve.
Okay, so the Purikoff pathway is like the emergency brake, and it selectively boosts the production of a manager, ATF4, who starts organizing the longer-term response.
Yeah, that's a good summary. It's the rapid response arm.
Okay, what's the next branch? You mentioned IRE1.
Yes, IRE1, which stands for inositol requiring enzyme 1. IRE1 is arguably the most evolutionarily conserved, meaning it's found in almost all eukaryotes from yeast to humans, and perhaps the most complex of the three sensors.
Older and more complex. Yep.
Okay, like PERK, when BP releases it, IRE1 dimerizes, but IRE1 has two enzymatic activities. It acts as a kinase, like PERK, but also, very unusually, it functions as an endonuclease.
An endonuclease. It cuts nucleic acids, like DNA or RNA.
It cuts RNA. Specifically, it targets one particular messenger RNA transcript inside the cytoplasm called XBP1 mRNA.
Wait, hang on. So this sensor protein, IRE1, when activated by stress in the ER, reaches out into the cytoplasm and performs surgery on an RNA molecule.
Precisely. It performs a highly specific, unconventional splicing of the XBP1 mRNA. It removes a small intron, a piece of the RNA.
Why? What does that do?
This splicing event shifts the reading frame of the mRNA, creating a new active protein product when it's translated. This product is called spliced XBP1, or XBP1s, and XBP1s is a highly potent transcription factor.
Ah, another manager molecule heading for the nucleus.
Exactly. This newly synthesized XBP1s translocates to the nucleus, binds to specific DNA sequences, and is responsible for activating two massive adaptive responses. This is like the cell urgently recruiting specialized cleaning and repair staff.
Okay, what are the two responses driven by XBP1s? First, XBP1s massively upregulates the production of genes encoding ER chaperones, like BP itself, and foldases, like PDI. This directly boosts the ER's native folding capacity. This is action 1, increase chaperones. More hands on deck for folding, makes sense. And the
Second, and equally important, XBP1 significantly boosts the machinery for ER-associated degradation, or ERAD.
ERAD, let's pause on that. That's the disposal system for bad proteins, right?
Yeah.
How does the UPR via XBP1s enhance this degradation?
Right. ERAD is the sophisticated pathway responsible for identifying hopelessly misfolded proteins within the ER, physically transporting them out of the ER lumen back into the cytoplasm, and then targeting them for destruction.
Okay.
XBP1s upregulates the genes for many key ERAD components. These include proteins that help recognize and extract the misfolded substrates, but maybe most importantly, enzymes called E3 ubiquitin ligases.
Ubiquitin ligases.
Yeah.
Ubiquitin is that tag for destruction.
Exactly. These E3 ligases are critical because they recognize the misfolded proteins as they emerge from the ER into the cytoplasm and tag them with chains of ubiquitin, that molecular kiss of death label we've talked about before.
And once it's tagged,
Once it's polyubiquitinated, the protein is immediately recognizable by the proteasome, that massive protein shredding complex in the cytoplasm. The proteasome grabs it and chops it into tiny pieces. So XBP1s, generated by IRE1's RNA splicing trick, ramps up both the folding assistance crew, chaperones, and speeds up the trash removal process, ERAD machinery, including the tagging enzymes, that's action 2, enhanced proteasomal degradation. You got it. So IRE1 increases the folding staff and accelerates the disposal of the irreparable rejects.
that's remarkable. A single sensor enzyme, IRE1, having these two huge downstream effects just by snipping one piece of RNA. Amazing. What about the third pillar, ATF6?
Right. The third sensor is ATF6, which stands for activating transcription factor 6. ATF6 is maybe the simplest conceptually. Like the others, it's kept inactive by binding to BP in the ER membrane when the cell is happy.
Okay.
Upon ER stress, BP lets go. But unlike PERK and IR1, which do their signaling right there at the ER membrane, ATF6 itself is actually a transmembrane protein that acts as a latent or proform transcription factor.
So it's not an enzyme like the others.
No. When released from BP, ATF6 doesn't signal from the ER. Instead, the entire ATF6 protein physically leaves the ER and travels through the cytoplasm to the Golgi apparatus.
The Golgi, the cell's post office and processing center.
Exactly. In the Golgi membrane, ATF6 encounters specific proteases enzymes that cut proteins called S1P and S2P. These proteases recognize ATF6 and cleave it, snipping off its N-terminal domain, the part that was sticking into the cytoplasm.
And that snipped off piece.
That snipped off piece is the active ATF6 transcription factor. It's now free to travel to the nucleus.
So it takes a trip to the Golgi to get activated before heading to the nucleus.
Correct. Once in the nucleus, this active ATF6 fragment binds to DNA and drives the expression of its own set of target genes. Interestingly, these overlap significantly with IRE1's target specifically. It strongly induces genes encoding more BiP and other critical ER chaperones and folding enzymes. So ATF6 primarily contributes to action 1, increase chaperones again. Primarily, yes. It acts as a sort of secondary, perhaps slightly slower, but sustained source of enhanced folding capacity, reinforcing the action started by IRE1-XBP1s.
Okay, let me try to summarize this incredible molecular orchestration. When misfolded proteins build up, PREC acts first, slamming the brakes on overall protein production via EI2, but selectively allowing ATF4 synthesis. IRE1 senses the stress, performs RNA surgery on XBP1 mRNA, creating XBP1s, which goes to the nucleus to ramp up chaperone production, action 1, and the ER disposal system, action 2. And ATF6 travels to the Golgi, gets cleaved, and its active fragment goes to the nucleus to provide another sustained boost to chaperone production, also action 1.
That's an excellent summary. Those three pathways, working together, represent the adaptive UPR, the cell's best effort to restore protein homeostasis.
It's a beautifully complex system. That strict, almost absolute quality control, though, it seems both necessary for preventing toxic buildup, but also immensely frustrating when you think about diseases where maybe a slightly flawed protein could still be useful.
Absolutely. And that strictness brings us to the pivotal transition. The cell is executing its triage, plan, boost, repair, enhanced disposal, cut supply. But what happens if the stress is just too overwhelming? Or too prolonged? Or the misfolded proteins are simply too sticky and abundant to clear out effectively?
Right. What happens when the cleanup fails? The cell crosses the Rubicon, doesn't it? It shifts from survival mode to self-destruction.
That is the critical point of no return. If the cytoprotective UPR response, everything we just described, cannot restore homeostasis within a certain time frame or below a certain stress threshold, the cell essentially recognizes that surviving with a chronically jammed ER full of potentially toxic, dysfunctional proteins is actually a greater threat to the whole organism than its own death.
So it initiates apoptosis.
It shifts its strategy actively towards apoptosis.
How does that switch happen? Is there a specific molecule that says, okay, plan A failed, initiate plan B, self-destruct?
There is. The transition is largely mediated by one specific, quite cytotoxic transcription factor that gets strongly induced under prolonged or severe ER stress. We mentioned it briefly as being downstream of the PR pathway via ATF4. It's called CHOP. CHP stands for CEBP homologous protein. You can think of it as the executioner's apprentice, or maybe the signalman who flags the train towards the apoptotic track when the repair track is blocked.
How does CHP act as this molecular switch for cell death?
Well, CHP expression is usually kept very low. It's weakly induced initially by ATF4 as part of the adaptive response, but it only reaches really high damaging levels when PERK activation is sustained and intense, indicating the stress isn't being resolved.
So chronic stress leads to high CHP levels.
Yes. And high levels of CHP do several things to push the cell towards death. It directly inhibits the expression of several crucial anti-apoptotic proteins, like members of the BCL2 family, which normally act as guardians protecting the cell from apoptosis.
It removes the protectors.
Exactly. And conversely, CHP is known to upregulate certain pro-apoptotic proteins. Plus, it contributes to the production of reactive oxygen species, ROS, which further damages the cell and amplifies the stress signals.
So sustained stress keeps PPOC active, which keeps ATF4 active, which eventually leads to maximum CHP expression. And CHOP then actively dismantles the cell's survival programs and promotes the death programs.
That's the essence of it. When high CHOP levels basically confirm the failure of adaptation, the cell activates the full cascade of cysteine proteases known as caspases.
Ah, the caspases, the executioners themselves.
They are. Once activated, caspases systematically dismantle the cell's essential structural components, chop up its DNA, and degrade key proteins in a controlled, contained manner. This leads to the characteristic features of programmed cell death, or apoptosis.
It's a clean suicide, right? To avoid inflammation.
Yes, the cell packages itself up neatly for disposal by phagocytes, preventing the potentially hazardous release of its internal contents, which would trigger damaging inflammation in the surrounding tissue. It's a tidy, self-sacrificing exit.
An elegant, if grim, biological failsafe. But, as we mentioned, this failure of the UPR to cope, leading to apoptosis, has disastrous consequences in many human diseases.
Absolutely. Let's zoom out now and apply these mechanisms directly to human pathology. The failures of ER quality control, or the consequences of its activation, contribute to a really shocking range of diseases. We can generally categorize them based on what happens to the misfolded protein. Is it destroyed when maybe it shouldn't be, leading to loss of function, or does it accumulate and cause direct toxicity, often triggering apoptosis? Let's take the first case. Destruction leading to loss of function.
A classic, tragic example here is cystic fibrosis, CF. The most common mutation-causing CF, called delta F508, results in a CFTR protein, a chloride channel that is only slightly misfolded.
Slightly.
Yes. Crucially, studies show this mutant protein, if it could just get to the cell surface, actually retains about 5% to maybe 15% of its normal chloride channel function. Not perfect, but potentially enough to lessen the disease severity significantly.
But it doesn't get there. No. Because the ER quality control system, the UPR and ERAD machinery, is incredibly hypervigilant. It recognizes even this slight misfolding in the ER. It flags the protein, tags it heavily with ubiquitin, and ensures its complete destruction via the proteasome before it ever gets a chance to leave the ER and reach the cell surface where it needs to function.
Wow. So the cell destroys a potentially useful, albeit imperfect, protein.
Exactly. The result is a near-total loss of CFTR function at the membrane, leading to the severe defects in chloride and water transport that characterize CF. The quality control system is, in this case, brutally absolute. Its strictness causes the disease phenotype.
It seems almost counterproductive, evolutionarily speaking, that such a strict all-or-nothing system would be favored. But I suppose, as you implied, the risk of having even slightly damaged, potentially aggregating proteins floating around was deemed worse than zero function.
That seems to be the evolutionary calculation, yes. Preventing protein aggregation, which can be highly toxic, appears to have been prioritized, even at the cost of losing potentially useful, partially active proteins in some cases. Okay, so that's CF loss of function due to overzealous destruction. Now, contrast that with the situation where the protein isn't efficiently destroyed but accumulates. Right. A condition that perfectly illustrates the dual pathology problems from both accumulation and functional loss is ABA1 antitrypsin deficiency, A1ATD.
Okay, what does alpha-1 antitrypsin normally do?
It's a crucial protease inhibitor, mainly produced in the liver, that circulates and protects tissues, especially the lungs, from damage by enzymes like neutrophil elastase.
Okay, so what goes wrong in deficiency?
In the common genetic forms of A1ATD, the protein is severely misfolded due to a mutation, and this particular misfolded protein has a strong tendency to polymerize. It links up with other copies of itself, forming long, sticky chains or aggregates right inside the ER of the liver cells, hepatocytes that make it.
So it gets trapped in the factory.
Exactly. It accumulates within the ER. Because it's trapped, it cannot be secreted properly into the bloodstream to reach its main site of action, the lungs.
And the consequence in the lungs.
The absence of A1AT protection in the lungs leads to unrestrained activity of proteases like elastase, which progressively destroy the lung tissue, causing emphysema. That's the loss of function part of the disease happening remotely.
But you said dual pathology. What happens in the liver where it's accumulating?
That's the other side. The massive accumulation of these misfolded A1AT polymers inside the hepatocytes triggers profound chronic ER stress in those liver cells.
The jam we talked about earlier.
A severe persistent jam. This prolonged, unresolved stress relentlessly activates the UPR, eventually maxing out the CHOP pathway we discussed. This leads to massive apoptosis of the liver cells themselves. Over time, this cell death causes liver inflammation, fibrosis, scarring, and can ultimately lead to cirrhosis and liver failure. Wow.
So A1ATD is a perfect storm. The lack of the protein causes lung disease, while the buildup of the faulty protein kills the liver cells that make it. Precisely. It beautifully illustrates both potential negative outcomes of protein misfolding. And finally, perhaps the area that gets the most public attention regarding protein misfolding: the neurodegenerative disorders. Yes, indeed. In many devastating neurodegenerative diseases, the core pathology is defined by the toxic accumulation and aggregation of specific, abnormally folded proteins within or around neurons.
Well, think of Alzheimer's disease, characterized by plaques of aggregated amyloid beta peptides outside neurons and tangles of misfolded tau protein inside neurons. Or Parkinson's disease, with its aggregates of alpha-synuclein forming Lewy bodies. Or prion diseases like Creutzfeldt-Jakob disease, CJD, caused by the aggregation of misfolded prion protein, PRPSC.
So aggregation is the common theme.
Yes. These protein aggregates, these plaques, tangles, fibrils, and Lewy bodies, they progressively accumulate and are believed to directly or indirectly exert toxic effects on neurons. They physically disrupt cellular processes, they can impair proteasome and lysosome function needed for clearance, and crucially, they place an immense burden on the ER's folding and quality control capacity.
Triggering ER stress in the neurons.
Exactly. While neurons, especially long-lived ones, normally operate with a high level of folding capacity, they typically postmitotic. They don't divide and can't easily be replaced if lost. The chronic, unrelenting ER stress caused by these persistent, often insoluble protein aggregates drives sustained activation of the UPR pathways, particularly PERK, and the downstream induction of SHAP. Leading directly to widespread neuronal apoptosis and the progressive loss of brain function that characterizes these diseases. The UPR, desperately trying to adapt and clear the mess, is ultimately overwhelmed and defeated by the sheer scale and persistence of the aggregation problem, forcing the vulnerable neurons to self-destruct via that SHA-mediated pathway.
It's a grim picture, connecting this intricate molecular response directly to such widespread and devastating human conditions.
It really underscores the importance of protein homeostasis for long-term cellular health, especially in non-dividing cells like neurons.
Okay, this understanding of the UPR pathways, PERK, IR1, ATF6, CHOP-TAG, and their roles in disease naturally leads to thinking about therapies. Can we manipulate this system?
That's precisely where a lot of cutting-edge research is focused now. If we understand the UPR not just as a consequence of stress, but as a set of sophisticated control switches.
PERK, IR1, ATF6 controlling adaptations, CHFP controlling death.
Exactly. If we see them as switches governing the cell's fate, then modern therapeutics shifts from just observing the damage to trying to actively manipulate these switches for therapeutic benefit.
This is the really exciting area, isn't it? The UPR offers this unique opportunity, this control panel, where we might be able to tip the cell deliberately toward either survival or death, depending on the disease context.
Precisely. And researchers are essentially employing two fundamentally different strategies based on that idea. First, there's the protective strategy.
Okay, protective, meaning keeping cells alive.
Yes. This approach is usually applied in scenarios where preserving cell survival is paramount. Think about neurodegenerative diseases where we want to save neurons, or maybe during ischemic injury like after a stroke or heart attack, where we want to help damaged but potentially viable cells recover.
So how would you protect them using the UPR?
The goal here is to enhance the adaptive, the cytoprotective arms of the UPR. If the problem is, say, misfolded protein aggregation in a neuron causing stress, we want to maximize the cell's own cleanup crew and repair capacity.
Who's the good guys?
Right. This involves research into developing small molecules that might act as chemical chaperones, drugs that help proteins fold correctly, or finding compounds that specifically enhance, say, IRE1's splicing of XBP1, thereby increasing ERAD capacity and chaperone production selectively.
So help the cell help itself better.
Essentially, yes. By optimizing the cell's natural defense mechanisms, the hope is to keep these vulnerable cells, like neurons, from crossing that CHOP-mediated death threshold. Give them a better fighting chance to manage the toxic protein load and survive longer.
So in Alzheimer's, for instance, we'd want the UPR to be the most efficient repair and disposal crew it can possibly be.
That's the protective strategy goal. But, as you hinted, cancer presents the exact opposite challenge, leading to the second approach.
The destructive strategy.
Right. We now understand that many solid tumor cancer cells, especially those growing rapidly in the often harsh tumor microenvironment, which can be nutrient-poor and hypoxic, low oxygen, are actually under constant, significant low-level ER stress just to survive and proliferate.
Ah, so they're already living close to the edge.
They often are, and they rely heavily on their adaptive UPR pathways, the PERK and IRA1 branches especially, to cope with this intrinsic stress. They've essentially co-opted the UPR's survival mechanisms to tolerate conditions that might kill a normal cell.
They're addicted to the stress response for survival. In many cases, yes. So the tactical objective for cancer therapy using this strategy is the opposite of the protective one. It's to inhibit those adaptive UPR branches, particularly IRE1 or PERK signaling, specifically within the tumor cells. Why?
Yeah.
Wouldn't that help them?
No, because they need that adaptive response to survive their high basal stress. If you chemically block their ability to engage those coping mechanisms, if you take away their crutch, you intentionally push them past their tolerance limit.
Ah, you sabotage their survival strategy.
Exactly. Because the cancer cell is already highly stressed and dependent on the UPR, inhibiting the adaptive response can shove it abruptly over the edge into massive, uncontrolled apoptosis via CHOP or other death pathways. You're essentially exploiting its high stress load and UPR dependency as its Achilles heel.
That's brilliantly tactical.
Yeah.
Leveraging the cell's own survival mechanism to induce its demise, based entirely on the context of the cell normal versus cancerous.
It's a very active area of drug development, looking for agents that can selectively modulate these UPR arms.
Okay, let's connect a few more dots before we wrap up. The ER is an organelle, a component within the cell, but it obviously doesn't function in total isolation. We need to revisit the connections between ER stress and other major cellular injury pathways we've discussed previously. Absolute crucial. The ER is intrinsically linked to at least two other major hubs of cellular crisis management: calcium regulation and oxidative stress, ROS. Let's start with calcium. How does ER stress mess with that?
Well, the ER isn't just a protein folding factory. It also serves as the cell's main intracellular reservoir for storing calcium ions, A2+. Calcium levels in the cytoplasm are normally kept extremely low, while the ER lumen is tacked with it.
Okay.
Calcium is essential for many ER functions, including the activity of some chaperones like calnexin and calreticulin. But free K2+ in the cytosol is also a very potent second messenger, tightly controlled.
And ER stress disrupts this control.
Severely. When a cell experiences significant injury or severe ER stress, the integrity of the ER membrane can be compromised, or specific channels might open inappropriately. The result is often a massive, uncontrolled leak of Ca2+ from the ER into the cytosol.
Raising cytosolic calcium levels dramatically.
Yes, and an abnormally elevated cytosolic K2+ concentration is highly damaging. It acts like a bull in a china shop. It activates a whole host of dangerous enzymes that are normally kept in check. Things like certain proteases, calpanes that degrade the cytoskeleton, phospholipases that damage cell membranes, and endonucleases that chop up DNA. This accelerates the process of irreversible cell injury and death.
A direct route to destruction.
And it gets worse. This excessive cytosolic Ki2+ doesn't just stay there. It often gets taken up by the adjacent mitochondria.
The powerhouses of the cell.
Right. But mitochondria aren't designed to handle that much calcium. Overloading them with Ki2+ triggers mitochondrial dysfunction, impairs ATP production further, and causes them to release their own pro-apoptotic factors.
Creating a vicious cycle.
A lethal feedback loop. ER stress causes calcium leak Calcium damages damage mitochondria, worsen energy crisis, and release death signals, plus can't buffer calcium anymore. This further exacerbates ER stress and pushes the cell definitively towards apoptosis. The ER and mitochondria are in constant communication, especially during stress.
It paints a picture of this internal spiral of toxicity. Okay, what about the link to reactive oxygen species, ROS, or oxidative stress? We already noted that changes in redox state can cause misfolding, but does ER stress itself generate more ROS?
It absolutely does, making it another dangerous feedback loop. The protein folding process itself, particularly the formation of those disulfide bonds we talked about, is actually a significant source of ROS generation even under normal conditions.
Cool.
The enzymes involved, like ARO1, transfer electrons to molecular oxygen as part of the bond-forming reaction, creating hydrogen peroxide, a type of ROS. Normally, the cell has antioxidant systems to handle this.
But under ER stress?
When ER is overwhelmed and folding is inefficient, these ROS-generating processes can go into overdrive. The enzymes responsible for disulfide bond formation might become backed up or function improperly, leading to misdirection of electrons and increased generation of oxygen-derived free radicals, more ROS.
And these ROS then cause more damage.
Yes. These free radicals cause oxidative modification of lipids in membranes, and critically, they can directly damage proteins, including causing them to misfold or aggregate. So ER stress causes ROS generation, and that ROS generation worsens protein misfolding and further exacerbates ER stress. It just keeps pushing the cell closer to activating CHOP and the death pathway.
So the ER isn't isolated at all. Its health is tightly coupled to calcium balance, mitochondrial function, and the cellular redox state. The UPR isn't just monitoring protein folding, it's really acting as a central hub, sensing the overall health and stress level of the cell.
That's a very accurate way to put it. This multifaceted nature, these feedback loops, it brings us back to something you mentioned earlier, the principle of vulnerability. Why do some cells succumb to ER stress more easily than others?
Right. This raises that important question. How can we predict which cells will survive and which will die, even if they seem to experience the same intensity of ER stress? Yeah. Well, the outcome depends heavily on several factors. The specific cell type, its basal folding load and initial UPR capacity, and its intrinsic resilience mechanisms. Plus, of course, the severity and duration of the stress. Okay, let's break that down. Cell type first.
We already touched on this. Highly secretory cells, professional protein producers like pancreatic beta cells making insulin, plasma cells making antibodies, hepatocytes making albumin, and A1AT are inherently more vulnerable.
Why? Because their assembly lines are always running near maximum capacity.
Exactly. Their ER is constantly working hard just to meet normal demands. So any additional stress, like the high insulin demand and metabolic syndrome, or a viral infection, or exposure to certain toxins, can rapidly push them over the edge into acute ER stress and failure. They have less reserve capacity.
Okay, so secretory cells are naturally more vulnerable.
Yeah.
What about resilience and timing?
Even within a single cell type, the duration and severity of the stress are absolutely critical determinants of fate. Acute transient ER stress, a brief overload, often results in a successful adaptive UPR response. The cell ramps up chaperones and ERAD via IRE1 and ATF6, slows translation via PERK temporarily, resolves the backlog, and survives.
Adaptation works.
Yes, but chronic unresolved stress, even if it's relatively low level, is much more dangerous. Why? Because it leads to that sustained activation of PERK, which leads to the sustained high expression of CHP. And as we established, high CHP is the primary driver towards apoptosis.
So time is a crucial factor. A quick hit might be survivable, but a long grind wears the cell down and flips the death switch.
Precisely. The cell's vulnerability is ultimately a function of how much time and resources it has to successfully activate the adaptive IRE1 and ATF6 pathways to repair the damage before sustained PERK activation tips the balance decisively towards CHP and death. It's this constant microscopic race against time playing out inside stress cells.
An incredibly dynamic balance. This deep dive has truly laid bare the elegance, the complexity, and sometimes the brutality of cellular quality control. The endoplasmic reticulum isn't just some passive structure. It's really the ultimate arbiter of protein function and, consequently, cellular fitness and fate.
Well put. So, just to summarize what we've unpacked today for everyone listening, when the ER's protein folding assembly line gets jammed, maybe due to high demand, genetic flaws in the proteins, environmental insults, or lack of energy. That condition is ER stress.
Right.
The cell immediately launches the unfolded protein response, UPR. This is a multi-pronged defense system using three key sensors, PERK, IR1, and ATF6. Their initial goal is adaptive. PERK hits the brakes on production. IR1 and ATF6 work to boost the folding helpers, chaperones, and enhance the trash disposal system, ERID, the cytoprotective phase.
Exactly. But if this protective response fails because the stress is too severe or too chronic, the sustained signaling, particularly from PERK, leads to high levels of the transcription factor CHOP. CHOP acts as the molecular switch, shutting down survival pathways and activating the caspase cascade, which leads to programmed cell death, apoptosis.
A shift from adaptation to elimination. And this is a universal biological process. It governs whether a cell lives or dies in response to protein folding problems. We've seen its signature fingerprints all over major diseases, the tragic loss of function in cystic fibrosis due to premature destruction, the combined organ toxicity and functional loss in alpha-1 antitrypsin deficiency, the relentless neurodegeneration driven by protein aggregates in Alzheimer's and Parkinson's, and the metabolic crisis underlying beta cell failure and diabetes. It's truly ubiquitous. And understanding these molecular sensors, PERK, IRD1, ATF6, CHOP, is what allows modern research to start thinking about strategically manipulating them, either boosting the survival signals in settings like neurodegeneration or deliberately pushing cancer cells that are addicted to the UPR over the edge into apoptosis.
The potential is enormous.
It is. And maybe to leave you and everyone listening with a final provocative thought, stemming particularly from those cases like cystic fibrosis, where the problem isn't aggregation but overzealous destruction.
Okay.
Well, since the ER quality control system is so absolute that it often sacrifices a protein like delta F508 CFTR that actually retains some partial function, could the next major therapeutic breakthrough involve something subtler than just trying to fix the misfolding itself?
What are you thinking?
Could we perhaps develop small molecules that don't necessarily make the protein fold perfectly, but instead act as temporary tunable ER gatekeepers or ERAD modulators? Could we find compounds that maybe temporarily inhibit the interaction between BP and the sensors just enough?
All right.
Or perhaps transiently interfere with specific E3 ligases involved in tagging that specific protein for ERAD?
So, trick the quality control system.
Essentially, yes. Allow that partially functional, slightly misfolded protein just enough time or opportunity to escape the ER's degradation machinery and reach the cell surface where it could still provide significant, maybe even life-altering benefit to the patient. Instead of fixing the protein, we'd be modulating the response to the protein.
Strategically sabotaging the cell's own quality control for the greater good of the organism. That's a really fascinating frontier for medicine, isn't it? Learning how and when to potentially override some of the cell's deepest, oldest safety mechanisms.
It poses interesting challenges, both technically and conceptually.
Indeed. Well, thank you for guiding us through that incredibly detailed exploration of the microscopic quality control systems that really do dictate health and disease at such a fundamental level.
My pleasure. It's a critical area.
And for everyone listening, now that you have the complete picture of ER stress and the unfolded protein response, you'll hopefully see the profound significance whenever that term protein misfolding enters the scientific conversation about chronic illness. It's not just a symptom, it's the trigger for this whole complex cellular drama. Thanks for joining us on the Deep Dive.